You know, if you look back at how we've tried to tackle the biggest biological challenges, I'm talking about chronic diseases, aging.
Right, the really complex stuff.
For decades, we were sort of stuck in what we now call reductionist thinking. We'd find one bad actor, one gene.
And try to fix just that one thing.
Exactly. And we learned a massive amount, but it always felt, well, incomplete. Because the human body isn't a broken machine, is it?
Not at all. It's a dynamic, deeply interconnected ecosystem.
Yeah.
And that's really the turning point we're seeing in the source material here.
This idea that we have to stop looking at isolated parts.
Absolutely. The mission now is holistic integration. It's about modeling the whole system. And for the first time, we actually have the tech stack to do it. Systems biology, multi-omics, and of course, the massive analytical power of AI.
And that sets the stage perfectly for our deep dive today. We want to understand this new framework that lets scientists finally map that biological complexity, especially around aging and senescence.
And this isn't just theory. This is about realizing the vision of 4P medicine.
Predictive, preventative, personalized, and participatory. The framework that makes it possible is called RamanOmics, and it bridges this historical gap between the molecular world of genes and the physical world of biochemistry.
Mm-hmm.
So before we dive into RamanOmics itself, we have to talk about that integration challenge. One of the papers uses this great analogy.
Ah, the city blueprint.
Yes. If the genome is the architectural blueprint for a city, the static instruction manual, what about all the dynamic processes? What tells you how the city is actually running right now?
That's where the multi-omics layers come in. The genome, like you said, is just the blueprint. It's only a tiny fraction of the story.
So what are the other layers?
Well, you start with epigenomics. Think of those as chemical annotations on the blueprint, little sticky notes that say, use this part now, or ignore this section.
Like DNA methylation.
Exactly. Then you have transcriptomics, which is the RNA. That's a snapshot of which instructions from the blueprint are actually being read at this very moment.
Okay, so we know which instructions are being read, but not what's being built.
Precisely. That's where you get proteomics, the actual protein machinery doing the work, and metabolomics, which is so crucial.
Why is that one so important?
Because it's the chemical fingerprint of what's happening. It's the exhaust fumes, the fuel consumption, the real-time chemistry of the city.
And it doesn't stop inside the body, does it? There's this huge external conversation happening.
A massive one. You've got the microbiome, trillions of microbial partners running our digestion and immunity. And then there's the exposome.
The exposome. I think this is such an underappreciated concept.
It's everything. It's the air you breathe, the water you drink, UV radiation, your diet, every single environmental exposure you've ever had.
So it's not just about your inherited risk. It's about this constant stream of inputs the body has to manage.
That's it. It's the context. It helps explain why one person with a risky gene gets sick and another doesn't. You have to look at their entire environmental history.
But that creates this astronomical challenge, right? We're talking millions of data points per person from labs, wearables, sensors.
It's an overwhelming amount of high-dimensional data and traditional stats. They just collapse. They can't handle it.
And this is where AI comes in. It's not just a tool anymore.
No, it's the essential master integrator. AI algorithms are uniquely good at spotting patterns across all these different messy data sets. Patterns a human could never see.
It finds the signal in the noise.
Exactly. It integrates everything, the exposome, psychosocial factors like stress, sleep, to build these incredibly rich models. And it doesn't just tell you what is different. It helps translate that complexity into actionable clinical advice. That's the engine for 4P medicine.
Okay, so this is where we get to the really interesting part. The systems biology approach, mapping all those omics, always had this one big problem.
The disconnect.
The disconnect between the molecular code, the genes, the instructions, and the physical reality of the cell, the actual chemistry and structure.
Right. The blueprint might say build a wall, but how do you know if the bricks are crumbling?
And that is the exact problem RamanOmics was built to solve. It maps the molecular conversations in context with the cell's physical state. So walk us through it. What are the components?
It's a three-part platform. First, you have things like single nucleus RNA sequencing to get a very precise molecular profile of each cell.
The what?
The what. Second, you use spatial transcriptomics like STARmap to know exactly where in the tissue those genes are active.
But where?
And third, the real innovation is label-free Raman imaging. This connects the molecular map to the physical chemistry.
Wait, so if we already have the RNA data and its location, why do we need another measurement? Why measure the chemical bonds? Isn't that redundant?
That is a fantastic question, and it gets to the limits of just looking at genes. Raman imaging measures the intrinsic physical properties of a cell
How so?
by measuring the vibrational energy of chemical bonds. Things like the saturation of a fat molecule or how a protein is folded.
And why is that so critical?
Because those biochemical changes can happen before a gene ever gets turned on or off, or they can happen completely independently. The cell's physical environment could be changing, and the RNA transcript alone would totally miss it.
Ah, I see. The body compensates physically before the genetic alarm bells go off.
For a long time, sometimes. Raman gives us that physical readout non-destructively right inside the intact tissue.
So you have a molecular map and a physical map. How do you merge them? How do you say this specific cell has these active genes and this exact chemical makeup?
It's all about registration. You need a common anchor point. In this study, they used the Raman signal from DNA
Okay.
and aligned it with the DAPI stain that's used in the transcriptomics. Once they're perfectly lined up, you can pair hundreds of chemical dimensions from the Raman data with hundreds of genes from STARmap for every single cell. It creates a unified multimodal identity for each cell.
Okay, so now we can really unpack what they found. They used this incredible framework to study aging and senescent cells in two very different mouse organs, the lung and the skin.
Right. And the first big insight is that aging looks very different depending on the tissue.
It's not one single process.
Not at all. In the lung, aging was primarily characterized by chronic, widespread immune activation. The whole tissue shifts towards this pro-inflammatory, pro-fibrotic state.
So it's basically scarring over time.
In a way, yes. Communication between the blood vessel cells goes down, but communication between immune cells ramps up. The classic pathways for scarring and rigidity, TGF-beta and collagen signaling were way up. The lung basically ages by getting inflamed and stiff.
And what about the skin?
A completely different story. Skin aging was all about metabolic decline, a failure of ion regulation. The key pathways that were elevated were all about structure and differentiation, like Notch and BMP signaling.
So the skin ages by sort of hardening its defenses while losing metabolic energy.
Exactly. The lung defaults to inflammation, the skin defaults to a kind of metabolic and structural failure. It's a powerful distinction.
Right. Okay, let's zoom in on the bad actors here. The senescent cells, the ones marked by P21 plus SOSA. What did their profiles look like in these two organs?
Their signatures perfectly mirrored what the whole tissue was doing. In the lung, the senescent cells were basically factories for fibrosis.
They were driving that scarring process?
They were upregulating genes tied to breaking down the local environment and causing scarring, and they also showed they were bad at handling lipids and oxidative stress.
And in the skin.
In the skin, they were all in on reinforcing that epidermal barrier. They were cranking up genes for keratinization, trying to make the wall more rigid.
But at a cost.
At a huge cost. Their ability to repair DNA was way down. So the lung cell promotes scarring, and the skin cell promotes this rigid barrier at the expense of any kind of regeneration.
And what about that weird finding about cell size? Tissues shrink with age, so you'd think the cells would too.
Right, that's the conventional wisdom. But they found the exact opposite. The senescent cells in both the skin and the lung were consistently and significantly larger than their healthy neighbors.
Larger. Why?
It's a key sign of their altered state. As they exit the cell cycle, they often accumulate more proteins and lipids. It's a physical change, a physical manifestation of senescence that something like Raman imaging is perfectly designed to capture.
So we've got the molecular differences down, but the real power of this RamanOmics approach comes from finding what's biochemically the same. What was the one chemical signature that popped up in senescent cells in both the lung and the skin?
This is the real aha moment in the data. The core finding was this incredibly consistent biochemical signature, a universal enrichment of a specific kind of fatty acid.
A branched chain fatty acid.
Branched-chain fatty acid linked profile. It robustly marked every single P21 plus senescent cell, no matter which tissue it was in.
Okay, let's unpack that for a second. What are branched chain fatty acids, or BCFAs, and what does it mean if they're building up?
So your normal fats are mostly straight chains. These BCFAs have little side branches on their carbon backbone, which is unusual. When Raman imaging picks up spectral peaks that correspond directly to these molecules, it's a huge clue.
A clue to what?
It points to a fundamental metabolic shift inside the cell. They're accumulating these specific lipids, which is often linked with that permanent growth arrest and changes to the cell membrane.
And this was backed up by other chemical changes too, right?
It was. They also saw a reduction in saccharide-associated peaks. So the cells were basically shifting away from using carbs for energy and were just accumulating lipids in this abnormal way.
That is the ultimate proof of concept, isn't it? That universal chemical shift was something you'd almost completely miss if you were only looking at the genes.
Totally invisible to genomics alone.
So did adding this chemical data actually help them identify senescent cells better? They talked about a multimodal barcode.
Absolutely. This is where it becomes a real tool for 4P medicine. When they combine the features, the Raman chemical profile plus the gene expression changes, their machine learning models got significantly more accurate.
How much more?
Their ability to pinpoint a senescent cell jumped by over 5%. That fusion of molecular and physical data created a much more robust RamanOmics barcode. It's a stronger diagnostic signature.
And they tested it in a real-world stress scenario, not just in slow, steady aging.
They did. They used a mouse wound healing model where we know senescence pops up temporarily as part of the repair process.
And did the barcode hold up?
Perfectly.
Yeah.
They saw those exact same lipid peaks, the BCFA signatures, along with the skin barrier genes, pop up strongly on day 3 after the injury. It proves these signatures are functionally relevant, not just some artifact of old age. So I think the fundamental takeaway here is really a philosophical shift. We're moving away from that reductionist view fighting disease one molecule at a time and toward cultivating resilient health in a patient's whole unique system.
And to do that, you need this kind of systemic wisdom, this integration of the molecular and the physical. The RamanOmics framework really feels like the blueprint for how to do that. It finally unifies the spatial molecular conversation with the actual physical chemical state of the cell. It bridges that gap. Which brings us back to the real-world application, back to 4P medicine. And here's the thought I want to leave with you, our listener. The Raman features in this study are non-destructive and label-free. So the big question is, could these signatures one day become real-time in vivo biomarkers? Imagine walking into a clinic, not for an invasive biopsy, but to have the chemical vibrations inside your tissues measured directly, to track tissue aging or to see if a senolytic therapy, a drug clearing out those bad cells, is actually working in real time. The study proved the signal is there. The next great challenge is getting that sensor technology to the bedside.